Hi, I'm Dr. Mike Milligan. I'm here with Dr. Garth Ehrlich from Drexel University, an expert in chronic bacterial pathogenesis. Garth, thanks so much for being with us.
It's wonderful to be here. Fantastic. Thanks for having me.
This is very interesting news. We're talking about the oral systemic link here, and Garth's research is so in tune with that. Tell us a little bit about some of the research that you're doing.
Some of the most important research that we're doing right now is establishing a link between periodontal bacteria and infections of the joint, whether they be periprosthetic joint infections, which means if you have an artificial joint it gets infected, or whether you have somebody with osteoarthritis and they get these periodontal pathogens in their knee.
Okay, so we know there's a strong association with periprosthetic joint infections. Tell me a little bit about that.
So periprosthetic joint infections are the most common reason that an artificial joint fails and has to be removed.
Right, which we don't want to do.
And we don't want to do because it's an extremely traumatic process for both the patient and the surgeon. They have to go in and break bone because the joint has been cemented into the bone.
Right.
But once you have a bacterial biofilm established on that joint, there is no way to eradicate it other than take the artificial joint out.
Right. And sometimes clinically you can't see this. It's subclinical.
That's exactly correct. There was only a small percentage of revision joint surgeries that were thought to be caused by bacteria. Right. And those were the ones that were caused by classic pathogens such as Staphylococcus aureus or certain streptococci. What we found when we started using this new molecular diagnostic was that essentially 100% of the revision arthroplasties resulted from bacterial biofilms. We just couldn't see them in the past with the tools that we had. These new tools allowed us to see that in the vast majority of cases there were bacterial organisms there that had come from the mouth.
Right. Now tell me about the osteoarthritis. What's the, we're quite sure that there is we don't know how much, but we're quite sure there is a significance of oral bacteria in osteoarthritis.
Well, that was even a bigger surprise because after we found the periodontal pathogens associated with the artificial joints, we thought, well, let's take a long shot and let's look at people who just have osteoarthritis. Right. And what we found was that the vast majority of people who have osteoarthritis have these periodontal pathogens in their joints. And so it raises the question, are these bacteria playing a causative or an exacerbatory role in osteoarthritis, which is one of the most common diseases, of an aging population?
If through dentistry and getting the mouth healthy, we can help the osteoarthritic patients, what a wonderful thing that would be for our patients.
That would be probably the most exciting finding that I would ever have in my career. It would impact a larger number of patients than anything else that we've ever done. And so if in the future, we can establish, that these periodontal pathogens are playing either an exacerbatory or causatory role in osteoarthritis, it provides a whole new, way of understanding and then ways of treating. You might be able to prevent or extend, the time that somebody, is artificial joint free. Because artificial joints only last so many years anyhow.
And your expectation through your research over time will be that this will come about, that you can show this?
We will be able to determine over the next, I would say, 5 years, whether the bacteria that we find in these osteoarthritic knees are associated with disease or whether they are just free riders, if you will.
Right.
It's an answerable question.
Right. Okay. Now, interesting to me, oftentimes you really don't see clinical periodontal disease in the mouth to a significant degree, but we know that there are bad pathogens in there. So similar to the knee, where you had the subclinical infections causing the problems in the joints, a lot of times this happens in the mouth. And so actually bacterial testing, like oral DNA and similar products, is something that's highly recommended now, because oftentimes when it clinically looks okay, there are really bad pathogens in there. Can you address that at all?
I couldn't agree with you more.
Okay.
The microbial burden is impossible to determine through visual inspection
Right.
or by culture. You have to use DNA-based methods in order to determine not only what bacteria are there, but the numbers of the bacteria, what the burden is, because that's absolutely critical. You have to not only be able to control the types of bacteria, but the numbers of bacteria. Because once you get the bacteria down below a certain threshold, you'll greatly reduce the risk for local disease in the mouth and certainly reduce the risk for systemic spread.
You said that a lot better than I did, Garth. So, thank you. So let's talk a little bit about biofilms. This is a big part of your research as well. Tell me about biofilms and how they change and why they're so hard for us in dentistry as well as throughout the body, and they're similar throughout, the ones in the mouth are similar to the body. Why they're so hard to eradicate.
Biofilms are essentially bacteria adopting a multicellular mode of life. So you're no longer dealing with a single-celled organism. You're dealing with a very sophisticated multicellular organism. The evolutionary pressures on a biofilm are at the multicellular level, not the single-cell level. What makes the biofilms in particular so resistant to eradication is that they are very highly resistant to our traditional antibiotics.
Right.
Because antibiotics primarily kill rapidly dividing cells. The bacteria in a biofilm, with the exception of the ones on the surface, are fairly quiescent in terms of division. They're metabolically active, but they are not divisionally active.
Right.
And so most of the antibiotics will not affect them. In addition, bacterial biofilms are not monoclonal. They're not made up of a single strain. They're polyclonal. It's like different types of tissues in an organ, if you will.
Right.
And each of the various strains that make up that biofilm have a different genetic makeup. And what makes a biofilm so potent is that the different strains constantly exchange genes through a process we call horizontal gene transfer.
Right.
So you're continually making new strains. So I like to think of a biofilm as like the mythical shapeshifters, if you will. Okay? So the immune system is constantly chasing a moving target.
Right.
And so by the time you get an immune response developed against a particular strain, it's morphed, and it's no longer the same thing. And so this makes the other reason why it's so hard to eradicate biofilms.
And we used to think this took a long time for these changes. Now we know it's very fast. Tell us about that.
The speed,
genetic changes.
Yeah, the speed with which horizontal gene transfer occurs in a biofilm is so much faster than we would have ever predicted. We did a study on a child who had chronic upper respiratory tract infections and chronic middle ear disease. And we showed that over a period of 7 months that one of the primary strains infecting the child of pneumococcus underwent 24 different transformation events.
Amazing.
That's about 4 a month.
Yeah.
Okay? So that by the time we looked at that strain 7 months later, 8% of its DNA had been replaced.
Amazing. Garth, is there anything else? I mean, we appreciate all your research and the significance of that as we look at the oral systemic link. Is there anything else that you would like to share with us about that, your research, the oral systemic link, before we close here?
All I'd like to say is that it is so important for the practitioners and the patients to work together to identify what bacteria you have and treat those bacteria. It will not only give you a better mouth, it will give you a better life.
Garth, thank you so much for all you do and
My
for
pleasure.
being with us today.
Thank you.